In this section, we're going to discuss tonicity. Tonicity describes the three conditions of solute concentration outside the cell membrane relative to the solute concentration inside the cell. Consider a cell surrounded by its semi-permeable cell membrane, surrounded by three different types of solution. That cell could be surrounded by a hypertonic solution, one that has a higher solute concentration than inside the cell. As a reminder, hyper means too much. A hypotonic solution would be one that has a lower solute concentration outside of the cell than inside of the cell. Hypo means too little. And an isotonic solution has a solute concentration equal to the inside of the cell. That prefix iso meaning equal. First, hypertonic solutions. Consider a solution of water and solute. The solution outside of the cell, outside here, is hypertonic when it has a higher solute concentration than the solution inside the cell. So in green, we have our solute molecules. So you see you have just a few solute molecules inside the cell and a lot of water. Outside the cell, you just have a little bit of water and a lot of solute molecules. So highly concentrated solute molecules outside of the cell. When this happens, the net flow of water is out of the cell. So we need to get more water out to that higher solute concentration. And when that happens, the cell will shrink and shrivel. Hypotonic. The solution outside the cell is what we call hypotonic when it has a lower solute concentration than the solution inside the cell. So take a look here. Inside the cell, there are many green solute molecules and fewer water molecules. You can see outside of the cell, there's more water and less solute. So a lower solute concentration on the outside of the cell is called hypotonic. When that happens, water will flow into the cell. Water flows in the direction of the higher solute concentration, so toward the higher solute concentration. So water will flow into the cell and the cell will swell. It will get bigger. An animal cell in a hypotonic solution, because it doesn't have those rigid cell walls, can swell too much. And when that happens, a cell will lyse or break, as you see here, and then the contents obviously come rushing out. Isotonic. The solution outside the cell is isotonic when it has the same solute concentration as the solution inside the cell. So you can see here, inside and outside the cell, we have equal numbers of solute molecules and water molecules, making an isotonic solution. In this case, there is no net flow of water. So water is flowing, but at the same rate in both directions. All right, for a little help remembering these different solutions, if a solution A has a higher solute concentration than solution B, solution A is hypertonic relative to solution B. Hyper meaning more, tonic meaning solute. So solution A has more solute relative to solution B. If solution A has lower solute concentration than solution B, solution A is hypotonic relative to B, where hypo means less, tonic means solute. Isotonic solutions have equal solute concentrations, iso meaning the same and tonic meaning solute. Remember that here we are differentiating solution A from solution B, but solution A could just be the environment outside the cell. Solution B could be the environment inside the cell. So use that to help you as you're deciding. All right, so here's a little table or chart to help you remember these different types of tonicity. So if the cell is in a solution that is hypertonic, that means that the greater solute concentration is outside and the greater water concentration is inside. The net direction then will be from the inside out, and you can see diagrammed here by these green arrows The net flow of water is from the inside out. There is water that moves in, right? Because we've got this dynamic flow of water in both directions, but the net flow will be from the inside out. And when that happens, it can cause the cell to shrivel. If the cell is in a solution that is hypotonic, then the greater solute concentration is inside the cell. The greater water concentration is outside. And the net direction of the water will be towards the greater solute concentration, so from the outside inside. So you can see the net flow of water from the outside in by these yellow arrows, and that can cause the cells to swell, or that will cause the cells to swell and can cause them to lyse. If the cell is in a solution that is isotonic, that means there is no greater solute or greater water concentration. They're the same, both inside and outside of the cell. And the net direction, there is no net direction of the water. Water is equally flowing in and out of the cell. Tonicity and plants. Plant cells have cell walls and permanent central vacuoles, which lead to different behaviors in hypotonic and hypertonic solutions. Water is stored in the central vacuole, and the size of the vacuole changes according to how much water is available to the cell. The rigid cell wall resists changes in cell size. The plant cell will swell only as much as the cell wall allows and will not burst in hypotonic solutions. The cell wall will not collapse in hypertonic solutions. A turgid plant cell is one that is full of water. It is swollen and cannot take in more water due to the pressure applied by the cell wall. A flaccid plant cell is limp and lacks turgor. Water has flowed out of the cell and it needs water. A plant with flaccid cells will appear droopy. If a plant cell loses too much water, the central vacuole is permanently damaged, the cell membrane is permanently damaged, and the cell is plasmolized. It will never recover. The higher the solute concentration in the solution, the smaller the central vacuole and the less turgid the cell. The cell wall resists changes in shape. So you can see in this hypertonic solution, water goes rushing out. We don't have enough water and we end up with a plasmolized cell. In an isotonic solution, we have a flaccid cell with this normal central vacuole here where we have this dynamic input and output of water. And in a hypotonic solution, water will rush in and we'll end up with a turgid cell with a very large central vacuole. So the higher the solute concentration in the solution, the smaller the central vacuole. So look at how the central vacuole gets smaller as the amount of solute on the outside of the cell increases. Freshwater weeds are helpful models of tonicity when viewed under a microscope. They provide an easy way to see cell walls and chloroplasts. Other structures are not visible. In the photomicrograph on the left, the chloroplasts are gathered around the perimeter of the cells, near the cell walls. You can see them here. This is because the cell's central vacuoles are filled with water, filling most of the volume of the cells, pushing the organelles to the perimeter. For comparison, the photomicrograph on the right here was taken of the same cells in salt water. Water has diffused out of the central vacuoles and out of the cells into the hypertonic environment. The location of the chloroplasts shows us that the cell membranes have pulled away from the rigid cell walls due to the shrinking central vacuoles. Shadows created by the cell membrane are visible. You can see them here. Models of semi-permeable membranes are used to study osmosis in the lab. Dialysis tubing is made of a transparent, flexible plastic compound that has microscopic pores. The diameter of the pores allows small molecules such as water, glucose, and ions to pass through. Sucrose, a disaccharide, is too large to pass through the pores. Therefore, dialysis tubing is a model of a semi-permeable membrane based solely on solute size. A dialysis bag is a section of dialysis tubing closed at both ends, which you can see in the image. A dialysis bag is used as a model of cell osmosis of a cell in osmosis experiments. In laboratory studies of osmosis and diffusion, a dialysis bag in a beaker of a solution can function as a model of a cell in its environment. Dialysis bags are made of sections of cellulose acetate dialysis tubing, which is a long tube of a semi-permeable membrane. A short section of tube can be closed off at one end, filled with solution, closed at the other end, and put into a filled beaker. For a typical osmosis experiment, dialysis bags are filled with different concentrations of sucrose and placed in beakers holding solutions with different concentrations of sucrose. Water will pass through the semi-permeable membrane, but the sucrose will not because the pores in the membrane are too small. If osmosis moves water from the bag into the beaker, the bag shrinks, decreasing in volume and mass. If osmosis moves water into the bag, it will swell and increase in volume and mass. Like plant cell walls, dialysis tubing is made of cellulose and will not pop under turgor pressure. Example 1, tonicity. State the net direction of osmosis and explain your reasoning. So inside of the dialysis bag that we're using as our semi-permeable membrane, we have a 1% sucrose solution, and on the outside we have a 0.2% sucrose solution. So what will happen? In what direction will the water flow? At 1% sucrose, the dialysis bag is hypertonic relative to the solution of the beaker. So water will flow in the direction of the higher solute concentration. So from the beaker into the dialysis bag until the bag becomes turgid. Example 2, tonicity. State the net direction of osmosis and explain your reasoning. So in this case, we have the dialysis bag with 0.1% sucrose solution, and outside in the beaker, a higher 0.5% sucrose solution. All right, in this case, the dialysis bag has a lower concentration than the beaker outside. So the dialysis bag is hypotonic relative to the solution in the beaker. Water will flow from the dialysis bag into the beaker until equilibrium is reached. And in that case, the dialysis bag will shrivel. Medical dialysis. The concept of osmosis is especially important for understanding kidney function. Dialysis tubing was developed to treat patients with kidney failure. One function of kidneys is to transport water and ions between blood and urine, maintaining the osmotic balance of the blood. When kidneys function well, wastes are disposed of via urine. Blood has a solute concentration of about 0.154 molar, and a healthy blood pressure is maintained. When kidneys fail, they do not efficiently remove wastes and excess water or maintain healthy blood pressure. Useful sugars and proteins may be lost in urine. Running blood through dialysis tubing in a dialysis machine rebalances the ions, nutrients, and water in the blood when kidneys failure. There are two types of dialysis: peritoneal dialysis, which is not being described, and hemodialysis, which is described on the next slide. A patient undergoing hemodialysis must have their blood dialyzed for a few hours 3 times per week. Blood is removed from circulation through a needle, run through a dialyzer, pictured, and then returned to the patient's circulation. Inside the dialyzer, blood is pumped through dialysis tubing that is surrounded by dialysate, a solution with perfectly balanced ions. The semipermeable membrane of the dialysis tubing acts as a filter, mimicking nephrons, the functional units of kidneys.